We're going to go to our next speaker. That's Dr. Jenny Molloy, who is a research associate at the University of Cambridge. So she's a chemical engineer at the Biotechnology University, developing open source tools for sustainable and equitable bioeconomy. Since graduating from the University of Oxford in 2015, the Doctor of Philosophy and Zoology is focused on genetic control of dengue mosquitoes. Jenny has founded 4 social enterprises, making open source tools more accessible to researchers and building community to support their use. As Executive Director of Beneficial Bio, a UK-based nonprofit, Jenny leads a team spread across 4 continents with hubs in Cameroon and Ghana to produce affordable reagents that accelerate biological research and maximize the impact of biotechnology for the benefit of global health in our environment. Jenny is currently chair of local production for diagnostics working group, which spun out from the UN Technology Access Partnership, also in 2020-2021 fellow of World Economic Forum, Future Council of Authentic Biology. Let's welcome Dr. Jenny Molloy.
Thank you very much. I hope you can hear me well. My colleague Stefan is actually going to introduce the first few slides. So if he's at the podium, I can start moving the presentation forward. And maybe he can say a couple of words of introduction to himself as well, as I was so eloquently introduced already.
Good morning, everyone. So my name is Stefan Fadanka. I'm part of Beneficial Bio Research Manager. I'm also the Executive Director of the Molloy Lab. And I'm very proud to also be one of the fellows for the Emerging Leaders in Synthetic Biology in Africa. So, thank you. So I'm here to present the work we are doing with Beneficial Bio with Dr. Jenny Molloy as well. I don't know if we can see the slides. Okay. So first of all, biology is the future of sustainable manufacturing. So that we know, but that feature is already here. It's just that it's not evenly distributed. We know that for many years, Africa has been quite limited in the contribution to research and science in general. So next slide, please. So the real problem we are trying to solve as part of Beneficial Bio is that innovation in biotechnology is stifled and prevented by scarcity of the tools to use to perform the experiment we do on a daily basis, and also to produce the products that we need to conduct our research. So the solution really that we want to bring in is to decentralize the means of production through distributed biomanufacturing, and also to create an abundance of tools that enable researchers to take the risk in discoveries and innovation. This is very important because in order to acquire new skills, in order to do the work we do, we need to fail. It's a trial and error process. So in order to fail, we also need the reagent and tools to be able to keep working, keep improving all the time. So really, that's why we are trying to bring in as a solution. So access to reagent through local production in Cameroon. So that's the first step we did. In order to speed up availability, reduce the scarcity issue of reagent, our solution is local production. Local production brings in a lot of advantages, such as control on the production cost of this reagent, and also availability of having access to the reagent in a timely manner. So what we did, we do a lot of activities. We produce locally produced enzymes. Currently, we work a lot on cell-based system, expressing protein using BL21 E. coli cells. But we are currently working on cell-free protein expression system as well. We do a lot of training and capacity building activities. Work a lot in collaboration with local and international research institution as well. So we also do a lot of hardware prototyping because the reagent access problem or ability to do research in general is prevented by the access of reagent, but also access of equipment. So in our company, we are trying to do some local prototyping of the tools and equipment that are needed for local enzyme manufacturing. So we do production. Our focus now is mostly DNA polymerases, but we also produce a lot of other reagents, such as refusion enzymes, and we are currently working on it. And we do a lot of business. So with the technologies that we use to produce the reagent, we need to make sure that they are available to the people who need them. So it's important to communicate a lot about what we do, and also to let people know that these solutions, these tools are available to them. So basically in the picture we can see behind, we can see the lab, the facility. We pioneer local enzyme biomanufacturing in Cameroon. Now we are able to do the local production. We have a fully equipped lab with R&D, sales and marketing and production department. Next slide, please. We mentioned producing enzyme for molecular biology research. That's what we do. We also use a lot of, as you see, we can see the process. We can see some example of the hardware tool that we locally developed. The one just beside my picture is the shaker incubator. We use that a lot in the protein production process. As I mentioned, we are using currently a cell-based system where we grow and use E. coli bacteria to produce the protein. And then we have the capacity to do quality control checks of produced proteins. We can see an example of a gel that were performed using our reagents. So next slide, please. So products, we also have products that are locally available, and then we make sure these reagents, as I mentioned, are available to researchers in Cameroon and elsewhere. We have an online platform where people can get information about the products, their price, everything. And we also prioritize a lot now and provide specialized tailor-made services such as mobile money payments and specialized customer service. Now meeting market needs. So now we can offer to our customers more affordable ambient temperature, and we can have researchers are free to have the reagent in less than 3 days when they are based in Cameroon, and not exceeding 8 weeks elsewhere in the world. So it's very important to mention here that as a company, we value a lot addressing environmental issues. So most of our products follow, we make sure to use clean technologies in the products we use. Next slide, please. And then, so I'll just give the floor to Jenny to keep up with the presentation. Are you here, Jenny?
There's a delay. Hopefully you can hear me. I think the Zoom can hear me. Hopefully the room can hear me as well.
Thank you. Yes,
great. Thanks,
Hear you.
Wonderful. So so I'm here representing Open Bioeconomy Lab at the University of Cambridge, and we've collaborated with the Molloy Lab and sort of co-established Beneficial Bio to translate research that my group is doing here at the university out into the real world. And one thing that makes that transition very seamless is that we base all of our research on reducing open technologies. Sometimes we've generated open source protocols, other times we've used protocols or technologies that were patented, but the patent is now expired. So many of the enzymes that we produce are built from an open source DNA collection, the Open Enzyme Collection, which contains over 60 enzymes and other useful coding sequences and proteins. If you're interested in obtaining this, it is available completely for free, shipping anywhere in the world from Free Genes at Stanford. So if you go to stanford.freegenes.org, you can order your free Open Enzyme Collection, and it will be delivered to you as a DNA toolkit that you can apply in your own labs. We've also designed it for example, producing enzymes that are useful in diagnostic research. So the Research and Diagnostics Collection has a whole bunch of selected and curated modules, including promoters, affinity tags, reporters, and many other useful bits of DNA for making enzymes specifically for isothermal amplification and for PCR and qPCR. Again, it's available for free from Free Genes, and it's been the kind of foundation of a number of activities, including we're currently developing a UK-Africa synthetic biology and diagnostics course called Bioengine, which will kind of use some of these tools. And really, when we're developing technology, we co-develop between Cameroon, the UK, and also a number of other collaborators in Latin America and across Africa. So we really try and focus on the needs, so ambient temperature storage, for example. Stefan already mentioned environmentally friendly, non-toxic, readily accessible chemicals, and really making the biology do a lot of the heavy lifting. So these are just some examples of ambient temperature stored reagents. We work a lot with Sanchita Bardran and Andy Ellington at the University of Texas, Austin, who have an amazing system called cellular reagents, which is effectively unpurified molecular biology reagents, and that works really well. You can see here at PCR after 7 months at room temperature. As Stefan mentioned, we also work on cell-free systems, but we're running out of time and probably over time, so I won't dwell on that. But just to say that we're always trying to kind of reduce the number of steps in the protocol. So we use auto-induction, actually using milk, so dried milk, fresh milk, whey, lactose, all of it induces our proteins. We're also looking at auto-hydrolysis to remove the need for DNA treatment. And we've come up with a number of protocols that kind of take steps out of the protein production process. We heard a lot about shaking incubators from Tina and Vanzi. So we've actually just kind of worked around that by inducing protein production on plates. And so you can see here a couple of different examples, but we basically use an auto-induction agar media, grow the plates overnight, scrape the bacteria off of the plates and process them without having to really do much optical density monitoring or centrifugation at the end. So it means that you can really concentrate the biomass. You don't need a big centrifuge, you just need a small one. So we're trying to kind of come up with these ways of making the process easier, both from a kind of research perspective, but also from a bioproduction and commercial perspective as well. And hopefully this will go past. There we go. And yeah, from one plate, you can get thousands of PCR reactions worth of enzyme. And so lessons learned, we set up the biomanufacturing facility in about 18 months for approximately $150,000, starting from a room, no lab. Equitable partnership has really been key between myself, my group in Cambridge, Stefan and his team in Cameroon, and all of our other partners. We're always working on how we can best kind of collaborate better. You can focus innovation on solving problems in context and work with researchers, but it really is only one part of the puzzle in terms of solving the issue. So the product might be right, but we've still got to figure out logistics and training and all of the stuff that many speakers, including Inkwaba earlier, pointed to that are real challenges of doing biology. And finally, that we are seeking, and people are seeking us out to set up new bioproduction nodes and kind of join forces as a collaborative network of African-led businesses and also businesses led elsewhere. We're talking to people in Chile and Argentina and the Philippines at the moment to kind of have a network of businesses that can really support each other in this endeavour. Thank you very much for your time, and to the organisers for this excellent conference. It's really been an honour to present the work that we're doing.